Hi, my name is Katie.
I'm Bella.
And I'm Evan.
In today's video, we will be discussing the communication between plants and fungi. We often think of plants as solitary, but in fact, they are communicating underground with a network of fungi. We will be focusing on a type of mycorrhizal fungi called arbuscular mycorrhizal fungi. Arbuscular means a tiny tree-shaped fungal structure that grows in the intracellular and intercellular regions of the root. AMF colonizes the roots of the plant as shown in this drawing here. On the left is a plant without mycorrhizas, and on the right, there is a network of fungi that is formed around the root that represents the mycorrhizal network. For the life cycle of AMF are obligate biotrophs, meaning that they rely on an autotrophic host, in this case plants, to complete their life cycle and generate new spores. In the first step of its life cycle, the fungal spores germinate in the soil and begin to grow fungal threads called hyphae. In the presence of a host plant, exudates or chemical signals from the root stimulates increased hyphal branching to increase its chances of contact with the plant root. In the second step, when the fungus encounters the root surface, hyphae penetrates the cell wall and forms a hypomodium structure. This hypomodium structure serves as an anchor to allow the fungus to extend its hyphae into the root cells of the plant, in which arbuscular structures can form for nutrient exchange in step 3. Lastly, in step 4, the mycelium, a network of branching hyphae forms in the roots and the soil. There are extra-radical mycelium, which grows in the soil to deliver nutrients from the soil to the plant, while intra-radical mycelium grows within the roots to absorb carbon from host plant roots. AMF is extremely prevalent in nature. In fact, 80% of land plants have AMF associated with their roots that allow them to communicate with other plants.
In order to better understand the relationship between AMF and plants, we first have to understand what symbiosis is. Symbiosis refers to the relationship between two different organisms. The key difference between these relationships is the impact on one another. In this diagram, we can see positive beneficial relationships with a plus sign, negative relationships with a minus, and neutral relationships with a 0. Some examples include predation, competition, commensalism, amensalism, mutualism, and parasitism. To further unpack some examples, we can look at these 3. The first is mutualism, where both parties are benefiting. In this case, we see honeybees spreading pollen for flowers while also getting nectar for honey production. Secondly, we can look at commensalism, when one party is benefiting while the other is unaffected. In this case, we see whales completely unbothered by barnacles, while the barnacles are given access to food and a safe location to live. Lastly, we see parasitism, where one party is benefiting while the other party is negatively affected. In this case, the cat's skin is very irritated, but the tick is getting a meal. In the case of AMF in plants, we see mutualism, where both of the parties are benefiting. Here, the fungi is able to provide nutrients to the plant, such as phosphorus and nitrogen, as well as increasing the plant's water uptake. On the other hand, the fungi can gain carbohydrates, such as glucose, from the plant. It's a win-win.
The establishment of this symbiotic relationship is mediated by the phosphate starvation response. The plant and fungus exchange nutrients over the periarbuscular membrane, and this requires a special adaptation from both mycorrhizae recognize strigolactone hormones the plant produces in phosphate starvation and release a myc factor, which is recognized by the plant's membrane receptors. The key players mediating the PSR pathway are inorganic phosphate, PSR transcription factor, PHR, PSR1 binding site, P1BS, and the strigolactone hormones. Phosphate is an essential nutrient for plants, and its presence is a negative regulator of the PSR. PHR is a positive regulator of PSR and acts as a transcriptional promoter for genes with P1BS. Many different PSR genes have P1BS promoter regions, including phosphate transporters, phosphatases, and regulatory microRNAs. But most important for mycorrhizal symbiosis are genes promoting the production of strigolactones. At high phosphate concentrations, plants produce inositol phosphates. Inositol phosphates are the natural phosphate sensor for the phosphate starvation response. They act as a cofactor for SPX domain-containing proteins. When active, SPX proteins bind and sequester PHR, preventing P1BS binding and PSR gene expression. Strigolactone synthesis is suppressed and mycorrhizal symbiosis does not occur. When phosphate concentrations are low, inositol phosphates can't be synthesized. Without inositol phosphate, SPX proteins are unable to sequester PHR. PHR is then free to bind P1BS and promote transcription of PSR genes such as CCD8. CCD8 is an enzyme that catalyzes the synthesis of strigolactones from beta-carotene. Excess strigolactones are released into the environment where they're recognized by mycorrhizae. The fungi release Mike factor in response to strigolactone detection. Mike factor is then recognized by the cell, allowing for hyphal infiltration and the establishment of the perialveolar membrane. Once this relationship is established, different plants can exchange nutrients and signal chemicals across the hyphal network connecting them. The exact mechanism of nutrient and signal transportation is unknown, but there are multiple hypothesized mechanisms. In A, nutrients and chemical signals are transferred directly from the cytoplasm of the plant to the cytoplasm of the mycorrhiza, and vice versa. In B, signals flow apoplastically through the cell wall of the hyphae. Apoplastic transport could also take place through biofilms or with the aid of proton gradients. In C, molecules are transported through the interior of the hyphal cord, diffusing through water or air in the extracellular space.
These microsomal relationships can transport more than just nutrients. The networks operate as their little own hormone highway, allowing communication between different plants and even species. In a 2014 study on tomato plants, Song et al. Established that mycorrhizal networks can be used to warn neighboring plants about environmental threats. They placed donor plants infested with predatory insects near healthy receiver plants, and then measured the defense enzyme response in the healthy plants. The healthy plants expressed higher defensive enzyme levels than those without mycorrhizal connections. Here we can see an increased hormone response from the jasmonic acid from the infested plant to the healthy plant. Due to the hormone increase, there was also an upregulation of defensive enzymes, which would allow future protection from predators in the future. Now that we know how these relationships are formed, we consider how the evolutionary advantages of them. For the relationship between kin or the same species, there seems to be an evolutionary advantage to helping one another in order to increase the likelihood of these shared genes, making it into the next generation. When plants have shown communication across different species, this is far more likely to be a form of group selection, as a healthier forest is one that is more diverse.
Here's a summary of what we covered today. On the plant community level, there are different species of plants in an area that have a mycorrhizal network. On the plant physiology level, when a plant encounters predation by herbivores, plant stress signals are released through the soil. These signals are received by a nearby healthy plant through the mycorrhizal network, causing it to upregulate defensive enzymes to protect itself from potential predation. And on a cellular level, AMF exchanges nutrients like phosphate to the plant cell in exchange for sugars.